Yeah, thank you so much. I'm having such a great time at this meeting. Thanks for putting it together, and thanks for involving me in this meeting. And I'm going to make a bit of a transition going from gigantic data sets focusing on a whole bunch of viruses to my bit of simplistic overview of what I think about when I think about a virus infection. So a typical virus infection starts with a single viral particle entering its host cell, and then if the infection is successful, the virus is going to replicate, it's going to make new copies of its genome, lots and lots of its proteins to eventually spread from the initially infected cells to others. So if I want to understand what happens during an infection, I am essentially asking the question, how does the virus transition from one copy of its genome to many copies of its genomes? And I kind of refer to this as the one-to-many challenge of a virus. And it's actually kind of challenging to address this question because, like, you can imagine there may be different options for a virus in individual cells that can take place. And I'm just like only showing 4 options here. There may be situations in which the virus is not replicating at all. Some of you may refer to that as an abortive infection. There may be situations in which the virus is rapidly replicating, and there can be all of the intermediate options. But if we think about the tools that we're currently using to study viruses, they do not really cover all of those scenarios. So many tools require us to average a lot of cells all at once. And once we're doing bulk measurements, we're essentially just looking at an average that is heavily dominated by those rapidly replicating viruses. Or a lot of tools don't have the resolution to see those events in which there's not a lot of things happening, seemingly. And finally, a lot of assays require us to freeze the cells all at once at a single time point. We need to fix the cells, we need to lyse them, or we need to do all other kinds of horrible things to them, which take away the opportunity to follow the infection over time. And even worse, in this one example, it may seem as if those 3 scenarios are all doing the same thing because we froze them at the time point that they are seemingly the same. Whereas if we follow these individual cells over time, we can see that very different things are happening in those cells. So to kind of overcome these kind of limitations, I want to share a couple of efforts to develop live cell single molecule imaging tools to follow infections in single cells from the very first moment of an infection onwards. And the first of this is using the picorna viruses as an example. And this is a collaboration from my time in the Tannenbaum lab together with Frank van Kuppenveld. The picorna virus, as many of you may know, is a small positive sensed RNA molecule. For the purpose of this talk, this is what you need to know about it. The infection starts with the release of the genome into the cytoplasm, which is then translated by ribosomes of the host cell, resulting in the production of a polyprotein, which is being processed into, among other proteins, a polymerase. This polymerase then uses this incoming RNA molecule as a template to first make negative strand RNA, and then this negative strand of RNA serves as a template for more positive RNA synthesis. And those positive RNA molecules can then be translated to make more and more and more viral proteins. And some of these newly synthesized RNA molecules may even start to serve as a template for replication themselves. And then eventually, those newly synthesized RNA molecules and the viral proteins are assembled, and through lysis, the virus is spreading. So can we visualize this? Yes. So in order to visualize this, we took advantage of the SunTech system, and we engineered a picorna virus, Coxsackie virus B3, by introducing 5 copies of the SunTech peptide at the end terminus of the polyprotein. And then we infect STAP cells. STAP cells are cells that are stably expressing a GFP-coupled SunTech antibody. And this means that once the virus is getting translated, once the host ribosomes are going over the RNA molecule, those short peptides are being synthesized. And those peptides can be immediately bound by the GFP-coupled antibody, essentially resulting in the assembly of a lot of GFP fluorescence on the RNA molecule. And then many ribosomes can be translating the same RNA molecule simultaneously, so we get a very bright signal from this one RNA molecule. And that is exactly what we can see. We can see those really nice GFP foci that all correspond to individual RNA molecules. Well, this is a fixed experiment. I was promising you we can do this live. So this is a cell that is going to get infected. This is a STAP cell. It currently does not have any virus, and the infection is going to start in the bottom left part of the cell. There we go. There's the first RNA molecule. It's translating, turns off its translation for a while. It's replicating, and then we see this burst of newly synthesized RNA molecules, and eventually some of them are even replicating themselves as well. Well, and rather than just looking at all of those really fun movies, we can quantify in individual cells how is the viral infection progressing. And these are just 3 example cells in which I counted over time how many of those spots do I see. And I hope you can appreciate that even though the exact timing, onset, and number of molecules is very different between those 3 individual cells, they are all following a remarkable recurring pattern. And each of those patterns, or phases as we call them, reflects a unique stage in the viral life cycle. Phase 1, there's only a single RNA molecule. This is the incoming RNA from the viral particle that infected the cell, and the incoming genome is getting translated. Then translation turns off and the virus is replicating. Well, our assay requires the active translation of the virus, and translation and replication are mutually exclusive, so we don't see replication itself, but we see the product of replication. During phase 3, we see the release and translation of those newly synthesized RNA molecules. For reasons we don't entirely understand, there then is a plateau in the replication. There's no longer release of newly synthesized RNA molecules, but some of those newly synthesized RNA molecules serve as a template for more and more replication, because we see a second burst of newly synthesized RNA molecules in phase 5. So we now have the opportunity to follow in individual cells how the infection is progressing. And for example, we can quantify the timing and efficiency of each of the individual stages in the viral life cycle. And I'm here showing that as a Kaplan-Meier survival curve, meaning that in silico, all the cells have been aligned at the start of a certain phase, and as cells are exiting a certain phase and entering the next one, there's a drop in the graph. And from this type of analysis, we can find all kinds of interesting things. So for example, phase 1, translation of the first RNA, has this very long tail to the right side. In most infected cells, phase 1 is completed within 2 hours. However, there's this small fraction of cells in which it takes 4, 6, 8, or even 10 hours to complete the first translation. And largely as a consequence of that, or not, yeah, as a consequence of that, if we look at the assembly of all cells infected in a single experiment, and we look at any given time point, we will find cells in each of the individual phases, completely asynchronous. The other thing that we can see here has to do with phase 2, replication of the first viral RNA molecule. We can see that in a fraction of cells, 10 to 20% of the cells, the replication is unsuccessful. You can see this also in this example movie. The cell got infected, the RNA is translating, it turns off translation. It may very well try to replicate, but we never see any indication that this virus successfully replicated. We never see this like burst of phase 3 of newly synthesized RNA molecules, suggesting that phase 2 is a bottleneck for the virus, because we only see this for phase 2. So this is a really fun tool that we've been using to try to understand all kinds of things about picornaviruses, to try to understand how the virus is regulating its life cycle progression and how it interfaces with the host cell. And one example that I want to share, because I think many people have been talking about interferon, so I want to jump on that boat as well, is to try to understand what happens in a cell that has an activated immune response. What does that mean for the infection phases? Which of the infection phases are being hit by an activated immune response? So we treated the cells with interferon and then looked at each of those infection phases. And a very remarkable thing here is happening. We see a strong effect on specifically phase 2. Phase 2, replication of the first RNA molecule, and only phase 2. So previously, the replication failed in 10 to 20% of the infected cells, and now we see that the infection is abortive in 60% of the cells. Interestingly, the cells that do manage to complete phase 2 are seemingly uninfected in the remaining phases of the infection, suggesting that phase 2 is not only a bottleneck for the virus, it's also a one-time opportunity for our immune system to block the virus, or at least early in its infection stages. And I think this was a really fun experience for me and for everyone involved in this project to really try to dive into the positive RNA viruses. And we have since expanded this toolbox into different viruses, including positive and negative RNA viruses. For people interested in them, we gave them names that in retrospect we should have thought about better. The original system is called Virim. The next system is called Virim2. The next system is called Virim-NEC. So we kind of ran out of names. And I want to spend the remaining of my talk on my recent endeavors switching from RNA viruses into DNA viruses, which is very exciting, but also a very scary thing for someone that comes from the RNA field. And I also shifted away from the nomenclature of Virim and not just numbering it. So in my current endeavors, I'm using the herpes simplex virus 1 as a model for a DNA virus. And Neer already did a wonderful job introducing it. So the only thing I'm going to reiterate is that it is a DNA virus that replicates in the nucleus. And even though molecularly this is a very different virus, this concept of the virus starts with a single copy of its genome, and at the end of a successful infection, it has many copies of its genome is the same. So my quest for trying to understand what happens during an infection conceptually is still very much the same. I want to be able to look at the individual viruses infecting single cells and see it as soon as it enters the cell, and then to see how does it progress from this one to many copies of its genome. And learning from our past experiences, I wanted to make a couple of improvements. So first of all, even though Verum has been a very powerful tool, it requires engineering of the virus, which really limits the applicability towards other viruses. Secondly, next generation of Verum were based on generating antibodies, nanobodies, or peptides that specifically target viral ribonucleoproteins. And I also didn't want to have to do that for any next virus that I wanted to do. And then finally, I wanted to be able to use this assay on viruses that may have a so-called inactive state or latency. So I wanted my new assay, which I am now calling under the working title virus copy number counting, I wanted this to be independent of active translation or active viral gene expression. Well, unfortunately for me, there's a lot of tools in the DNA world, so I'm taking advantage of the Cas9 system. And specifically, I'm using a nucleus-dead Cas9. So the Cas9 is not going to cleave its target sequence. It's simply functioning as a vehicle for transferring fluorescence to my site of interest. Second of all, I'm expressing a pool of guide RNAs all directed against HSV1. And I want to stress here that usually when people talk about a pool or a library of guide RNAs, that means that you're hitting each cell with a unique guide RNA. In this particular case, all of those guides are co-expressed in the same cell. And what's going to happen is that as soon as the virus enters the nucleus, or as soon as the DNA is deposited into the nucleus, each of those guide RNAs in combination with a Cas9 can recruit a GFP fluorescence to the genome. And because we have this pool of guide RNAs, this allows for the accumulation of a lot of fluorescence all at once, already at the single molecule level, or a single genome count level. That's my one. And then as the virus is replicating, going from one to many, the size and intensity of this spot is going to increase. And we can then calculate how many copies of the genome are there. Well, and this is all very preliminary and still ongoing, but I'm so excited about it that I have to talk about it all the time. This is an example of how that looks like. In the top part, we have a cell that is uninfected. Unfortunately, this Cas9 has a tendency to accumulate in the nucleoli. So I'm going to ask you to ignore those very bright foci and instead focus on the dim foci. Bear with me. So there is this one spot, the dim foci, that is the viral genome. And then if the virus is replicating or reaching a late stage in infection, there's a lot of viral genome in the nucleus. And even in this case, there's already some nuclear reorganization taking place, and the viral DNA is already traveling to the nuclear envelope. Once again, we can even do this live. So this is actually the same example cell. The virus is going to enter in the left bottom corner. Again, I always orient my cells in the same way. And the virus enters in the left bottom corner. And there we see the genome. And then over time, this spot is going to increase in size and intensity, meaning the virus is replicating. So we can now do this in individual cells to quantify how many copies of the HSV genome are present at a given time point. And the very fun thing is we can not only do this for a single genome, but because we can track the genome, we can even look at multiple genomes in the same cell at the same time. So do a high MOI experiment. And to just show an example of a quantification of that, these are 4 genomes that all entered the same nucleus and then replicated or not. For example, site number 1 didn't seem to be replicating at all. Site number 2, it actually took a while for site number 2 to become present in the nucleus, and then it took even longer for it to even start replicating. Site number 4, it's replicating at a slow but steady pace. And finally, site number 3, it takes a while and then it shoots through the roof. And remember, all of those 4 sites are in the same nucleus. So any protein that is being produced by one of those genomes should be available to all of the genomes. yet not all of them are replicating, or not all of them are replicating at the same rate. And I think this provides a really fun opportunity to try to understand what is controlling the speed of virus replication. And I also specifically wanted to show this one example as kind of a PSA to everyone. When you do your high MOI experiments, you're masking all of those rare events, and you're gonna look at something that is probably dominated by the rare or frequent events of the genome that is rapidly performing its function. So to wrap up, when I think about a virus infection, and if I want to understand what happens during an infection, I'm translating that from how does the virus go from one to many? And tools like Virium or virus copy number counting would allow us now to focus on the early infection stages. How does the virus go from one to many, and how efficient does that happen? But also, when and how does that provide us as scientists or us as people with an immune system to interfere with that transition? And the other thing in the context of comparative virology I wanted to stress here is that it is really remarkable anytime I look at the data set of a virus experiment, be it different types of viruses or the same virus under different conditions. There's this enormous amount of heterogeneity. There is this wide variation in the exact start, efficiency, timing, number of molecules in the end between each of the viruses, even if we're talking about seemingly identical viruses, seemingly identical cells. And I really think this is an opportunity to start to dive in and to try to understand where does this come from and how can we take advantage of that. So I want to thank everyone that was involved in this project. The first part was from my time in the Tannenbaum lab. And my current efforts working on HSV1 is in the Euler-Yaniv lab together with all those wonderful people with whom I'm working on HSV. And with that, I'm happy to take any questions or to discuss how we can do live virus imaging on your virus of interest.